Hi, my name is Kerstin Lindblad Toh, and I'm from Uppsala University and the Broad Institute. I'm very excited to be the chair of this session on comparative genomics. We have 6 different talks, 1 long one and 5 shorter ones, where we are going to hear about a diverse set of species and a diverse set of projects. Because of the fact that we're running a little late, we are going to have all the talks coming together. First, Matthew Webster from Uppsala University, then Marco Corbo from UC Davis, then Garima Singh from Schenkenberg, then Shigehiro Kuraku from Riken, then Morgan Wirflin from Carnegie Mellon, and then Maribeth Goma from Ehime University. It would be very helpful if when you type in questions, you will also write the name of the person you're addressing your question to. So with that, take it away, Matt.
So I'd like to thank the organizers for inviting me to speak at this conference, where I'll talk about our work, where we've used population genomics to understand how species diverge and become isolated on a genomic level, focusing on Pyrobombus subgenus of bumblebees. So there are now a wealth of studies using genomics to study speciation, and the common observation is that new species often arise under conditions of gene flow. And under a commonly invoked model, regions of reduced gene flow arise and then expand through divergence hitchhiking. Such regions have been termed islands of speciation and contain variants involved in ecological specialization or genomic incompatibilities that are disadvantageous in the wrong genetic background, so gene flow is selected against. And a number of studies have used genome comparisons to search for such islands of speciation by looking at peaks of FST between related species in order to try and find barriers to gene flow that are important in speciation. But it's also been pointed out that processes such as background selection and genetic hitchhiking lead to peaks of high FST unrelated to speciation or gene flow. And examples in both of these phenomena have been found in many taxa, including both the genetic basis of genomic incompatibilities and genes underlying ecological specialization. But also a number of studies have shown, have identified genomic islands that are involved in, occur in regions of low recombination that are more consistent with being these incidental islands. So if we compare these 2 models, islands of speciation formed by differential gene flow and those formed by link selection, both are expected to result in peaks of FST. But the main, when comparing genomes, but the main way to distinguish between these is by the elevated DXY, which is absolute divergence, which robustly identifies differential gene flow, but not incidental islands. So I've been interested in applying some of this theory to understand the processes governing species diversity in this set of species, which is the Pyrobombus subgenus of bumblebees, which are found in that subgenus, distributed across Arctic and alpine environments. So we first generated a genome assembly of the species Bombus sylvicola. That could be used to map sequences from the subgenus using Oxford Nanopore, 10x and a standard assembly approach. And we placed the scaffolds on a genetic map of Bombus terrestris and also identified repeats that were potentially associated with centromeres. We then collected a few hundred samples of this species from 7 locations across the Rocky Mountains in Colorado, mainly in order to learn about population structure and local adaptation. Much to our surprise, we actually identified 2 distinct clusters when we analyzed this data. Both clusters were observed in all of the 7 localities and had no clear morphological differences. And after extensive analysis with other published data sets, we inferred that one of these clusters belongs to a new species, which we call Bombus incognita. So this gives us the opportunity to study how divergence has occurred on the genomic level in two species that are found in sympatry and likely to have had the opportunity for gene flow during their evolution. In order to compare the potential effects of gene flow on genome divergence, we also used some published genome sequences from a couple of other related species that are believed to have diverged recently in allopatry without gene flow. And that's Bombus bifarius and vancouverensis, which are found across the Western USA. So now we can make a comparison of how genomes diverge in allopatry and sympatry. Here you can see a tree of the species showing the pairs that occur in allopatry and in sympatry. So what we can now do is to make 3 independent comparisons of genome divergence here between populations on distant locations from the same species. Here is the sympatric genome comparison, and the third is the allopatric genome comparison. And you can see that there are peaks of divergence across all of these genome scans, and I've highlighted the islands of divergence as regions above 100 kb in red. And what it's clear is that there are peaks in all of the comparisons, and that they occur in very similar locations. So if we then analyze the location of these genomic islands, we can find that they tend to associate with regions of low recombination and with the putative centromeric repeats that we identified. And this is typical of incidental islands that are caused by link selection. However, we also observe differences between patterns of divergence in sympatry and allopatry. Firstly, it's clear that the distribution of FST in sympatry is bimodal. And this indicates a kind of a distinct class of highly divergent regions that could potentially be regions that are resistant to gene flow. And that's not seen in the other comparisons that we made here. Next, when we use the DXY statistic, absolute divergence, we can see that it's clearly elevated in genomic islands that are found in sympatry, but reduced in those that are found in allopatry. And so if you recall, this is quite a robust signal of differential gene flow, and it's present in the sympatric, but not the allopatric comparison. So to sum up the patterns we observe, we find islands of divergence in the same genomic locations in both the allopatric and the sympatric comparison. And they're strongly associated with the centromeric regions and regions of low recombination. So this generally implicates link selection in their formation. But we also see differences between the two species comparisons in terms of elevated DXY in the genomic islands and a bimodal FST in the sympatric comparison. And this elevated DXY is a particularly robust signal of the presence of differential gene flow in the presence of barriers to gene flow. So this indicates that differential gene flow has been important in the evolution of in the sympatric comparison, indicating that these islands of speciation have been reinforced. So just to conclude, as I mentioned in the beginning, there are 2 main explanations for these islands of divergence, either regions that are resistant to gene flow, or those that are found in regions of low recombination and are generated due to link selection. And I think our results suggest a way to synthesize these views, where we find evidence of islands that are shaped by both forces, whereby link selection can be important in forming genomic islands and facilitate the buildup of incompatibilities that then form barriers to gene flow in sympatry. And these findings are also consistent with some other recent studies in other taxa that indicate that there's an interaction between gene flow and recombination, suggesting that species barriers are polygenic and that regions of low recombination are more resistant to introgression. So genomic incompatibilities may be widespread and polygenic, and they tend to accumulate in regions of low recombination. So with that, I would just like to thank the people involved in this project. It's mainly been driven by a great postdoc in my lab, Matthew Christmas, with a lot of bioinformatics support from SciLife Lab in Uppsala, and fieldwork done in collaboration with Nikki Miller-Strutman and Jenny Gieb. I'd just like to leave you with this one final conclusion slide. Thank you for your attention.
Hello to everyone, I'm Marco Corbo, and today I'm going to talk about the evolution of chromatin conformation in carnivores. The genome is organized in three-dimensional layers, so we wonder whether the three-dimensional structure of chromosome is conserved between species. To answer to this question, we wonder whether IC maps can be used to identify homologous chromosomes in different species of the same family, whether chromosome rearrangements can affect chromosome conformation, and whether chromatin structure and compartments are conserved at the ordinal level. So we use genome assemblies and IC datasets generated by a standardized pipeline with peripheral bulk leukocytes provided by the DNA2 consortium. And we use three main approaches. The first one is a pairwise genome alignment using LASD. A second one is a direct IC map comparison to do pattern analysis. And then the third is the comparative eigenvector analysis of the IC maps with the values left over on the cat as reference genome to distinguish between the two compartments NB, which correspond to euchromatin and heterochromatin. So as part of the result, we show the analysis done on 5 different species of the Felides family, knowing that they have a stable karyotype and the same number of chromosomes. We analyze 2 different, in this example, homologs of cat chromosomes D3 and in1. And as you can observe by the genome alignment, the chromosomes are almost completely conserved, besides just small few inversion in few species. But when we analyze the IC maps, we were able to detect 2 different and interesting things. The first one is that every single chromosome has a specific and unique pattern. And second, and most important, that these patterns in this chromosome share the same chromatin conformation when we analyze different species. In this particular case, when puma, tiger, leopard, cheetah, and colored leopard were analyzed, they shared 19 out of 19 chromosomes with conserved chromatin conformation and with a unique pattern and conformation. On the right, we have the analysis on the eigenvector values, which were lifted over on the cat genome. And also here, through this quantitative analysis, we can observe that patterns and compartments are completely overlaid and shared. Then we moved to a different family with a different karyotype, with 3 species having a different chromosome number. In this example, we reported the analysis done on a C-scaffold 16 of the red fox, which corresponds to 3 different chromosomes in the dingo and the wild dog. When we analyzed the IC maps pattern, we could have identified that every single chromosome has a specific and unique pattern, which is shared between the 2 dogs, but also is shared in the red fox chromosome, which is fused, confirming that there is a conserved chromatin conformation between the 3 species, although the fusion is event. And then we detect that most of the fission and fused chromosome have an altered chromatin conformation. And then we expanded the analysis at the order level of the carnivores by including 3 species of the bird family. And here we show the analysis done on the homologs of cat chromosome 83. By looking at the genome alignment, we can observe an inversion which corresponds to the short arm of the cat chromosome 3 in 3 canids. which here is highlighted by this pink fragment. When we analyze the IC-map patterns, we could identify that this inversion, which is highlighted here by the green box, it's similar but inverted and specular respect to the non-inverted region of the other species. When we compare the long arm of the cat chromosome E3, which correspond to the blue box, we could observe that there is a complete chromatin sharing between all the different species. So we moved to the eigenvector analysis to access and detect whether this inversion could affect the status of the B compartment. But after overlaying every single species, here highlighted by different lines of different colors to the cat genome, we could observe that there was no switch between A or B in the inverted region in the gains. So we could say that in conclusion, chromatin conformation is conserved between species within the same family. So for species whose karyotype is unknown, AC data can be used to identify homologous chromosomes using a standardized pipeline and the same cell type. And then also the chromatin conformation at larger scale is not altered by inversion, fissions, or fusions within the carnivore order. Although possible changes of chromatin conformation near breakpoints can be still present at higher resolution, and we are ongoing on the analysis of this point and also to the relationship to the gene expression of phenotypic evolution. I would like to thank Professor Ernest Lewin, all the members of my lab, and all the authors of this work, and you all for your attention. Thank you.
Hello, everybody. I am Garima Singh. I work as a postdoc in the, sorry, I restart, sorry. My time didn't start.
No problem.
Okay. So, hello, everybody. I am Garima Singh. I work as a postdoc in Translational Biodiversity Genomics Center in Frankfurt, Germany. Today, I'm here to present my work on how biosynthetic gene clusters differ in high and low altitude populations of a lichen-forming fungus. Many organisms produce organic molecules called natural products or secondary metabolites, which serve important ecological functions related to abiotic stress tolerance, for instance, heat, cold, desiccation, and UV resistance. However, the mechanisms involved in the synthesis of these natural products are not very well understood, and the functions of many natural products remain unknown. In this study, we use a genomics approach to identify natural products which might be involved in stress tolerance. Specifically, we study biosynthetic gene clusters, which refers to the linear organization of genes involved in the synthesis of a natural compound. These clusters contain conserved genes which can be retrieved from the genomic data. We studied distribution of biosynthetic genes in natural populations, for instance, under different climatic conditions. And we look for the metabolites and genes which display climate-specific patterns. For this, we chose the lichen Umbilicaria pustulata as our study system because of its broad ecological distribution and its distribution across the mountains. And because its Mediterranean and cold temperate populations have already been shown to be differentiated into cold adapted and warm adapted ecotype. For this study, we chose 3 replicate gradients or 3 mountains, 1 in Italy, 2 in Spain. We collected 100 samples each from the top and from the cold temperate and Mediterranean region, so that overall we had 300 representatives of the cold adapted ecotype and 300 of the warm adapted ecotype respectively. We then used a population genomics approach to identify ecotype-specific clusters and ecotype-specific polymorphisms. Overall, we found 28 clusters, of which 25 were without any climate signals, and 3 clusters which displayed climate-related patterns. Out of these 3 clusters, 1 cluster was present only in the cold temperate regions and almost entirely absent from the samples which were collected from the Mediterranean region. And the two other clusters, they were functional only in the Mediterranean region, whereas in the cold temperate region, these were having deleterious mutations. So this is the cluster which is specific to the cold temperate ecotype. It's about 45 kb long and has 14 genes. And in the Mediterranean region, out of 14, 9 genes are almost entirely absent, and the other 4 are present only as fragments. Then out of the 2 cluster which showed ecotype-specific polymorphism, I am going to present here one example. This cluster is about 35 kb long, and we can see that in the cold temperate regions, the core gene of this cluster contains stop codons, 3 stop codons, and these stop codons are almost entirely restricted to the cold temperate regions only, whereas in the Mediterranean region, all the samples contain the functional copy of the putatively functional copy of the gene. To conclude, we can say that combining comparative genomics and population genomics is a suitable approach for us to discover genomic regions which might be involved in climate adaptation, and to identify biosynthetic gene clusters which might be associated with the natural products putatively mediating abiotic stress tolerance. With this, I would like to thank Translational Biodiversity Genomics Center for funding my research, and you all for your attention. Thank you very much.
My name is Shigehiro Kuraku. Let me introduce the project Squalomics. Squalomics aims to unveil the unique evolutionary trajectory of sharks and rays. It is mainly conducted by my lab and contributed by many people who provided samples, scientific inspirations, and biological solutions. Our collaboration involves quite a few aquariums in Japan that are enthusiastic and experienced with in-depth biological research. Here I list what makes working with sharks difficult. Especially for genomics, the large genome size has been the biggest obstacle. We published our interim results in 2018 for these species, including 2 relatively small egg-laying species, expecting them to be laboratory-friendly research targets in Asia Pacific. We reported in this paper, we reported quite a few gene-by-gene discoveries and genome-wide trends. Now we are going for the chromosome level, but karyotype repeats are extremely limited for sharks and rays. So we did karyotyping in-house. By referring to the obtained karyotype, we got chromosome scale scaffolds with the cost-saving Hi-C protocol, ICONIC, which again, we optimized in-house. This is a comparison among whale shark genome assemblies. Our latest assembly here highly resembles the karyotype, and we are further improving this assembly now. Likewise, we are working on several other species employing combinations of sequencing technologies that are suitable to the individual species. So far, we have got multiple tissue transcriptome data at least for 20 species. This lineup covers most of the entire higher order diversity, and so far we are working on 20 species, and we have got multiple tissue transcriptome data at least. This lineup covers most of the entire order level diversity, and we will continue to enrich the list. We have been working on various topics, including sex and vision. And now I visit these two topics in the middle in this today's talk. I guess many of you know Hox genes. Generally, Hox gene clusters of bony vertebrates are normally under structural constraint that packs the whole cluster within 100 kilobases or so, with only few repetitive elements inside. This condensed pattern is observed in A, B, and D clusters of sharks as well. Interestingly, this sizing of 100 kilobase seems to be maintained even in enlarged shark genomes. It was previously thought that sharks lack HOX-C genes. In our 2018 paper, we reported several HOX-C genes that are rapidly evolving and exhibiting a typical gene expression for HOX genes. In our latest HiC scaffold, the HOX-C genes are clustered on one end of a putative chromosome, and this region underwent massive repeat invasion, resulting in remarkable elongation of nearly twice as large. In conclusion, sharks have retained some HOX genes, possibly in a cluster, that escaped from the structural constraint. The last topic is homeostasis. The tree here shows the phylogeny of leptins, known as an obesity gene in mammals. We investigated various genes regulating homeostasis, and this diagram shows the early establishment of the components of what is called the gut-brain axis, and the secondary loss of some components in different lineages. Our transcriptome data of different tissues allowed us to identify similar expression profiles of these homeostatic regulators between the shark and the mammals, like the growth hormone in the pituitary, motilin gene in intestine. I cannot cover more in this short talk, but we will continue to work on various questions that are posed by the enlarged, slowly evolving genomes of sharks and rays. With these takeaways, I end my talk. Thank you for listening.
Well, hi, I'm Morgan Worthland, and today I'm going to tell you about the regulatory evolution of mammalian fine motor behavior. So fine motor control is at the very heart of what makes us human, from our ability to manipulate tools to our ability to speak to one another. It's really a defining feature of the human species. But how are complex behaviors like these encoded in our genetic material? Now, in the last 2 decades, we've seen massive expansion in the number of genomic resources available, ranging from the genome sequences themselves to transcriptomics that indicate which genes are being turned on or off in a given tissue. More recently, epigenomic data resources have also become available. And these are what really reveal the regulatory processes that underlie gene activity. So a truth that's beginning to emerge is that the evolution of complex traits is not governed by changes in protein coding genes themselves, which are largely conserved and comprise just 2% of our genetic material, but rather these gene regulatory epigenomic processes, which fill the other 98% of our genome. And the most powerful way to analyze these sequences is through a comparative evolutionary approach, as has been used to study the evolution of the human brain relative to chimpanzees, the evolution of limblessness in snakes, as well as the evolution of subterranean mammal phenotypes. To give you an example of why this comparative approach works, consider a shark, a dolphin, and an extinct ichthyosaur. Despite being completely unrelated organisms, these lineages converged on a highly similar phenotype due to similar environmental pressures and the demands of being an apex predator underwater. So by comparing them, we can start to identify fundamental principles underlying the evolution of being an underwater predator. Vocal learning, the ability to shape vocal behavior as a result of experience, as observed in human language and birdsong, is also another such convergently evolved trait, having evolved just a handful of times in the animal kingdom, in mammals as well as in birds. And as before, it is examining examples of convergence where vocal learning has evolved in unrelated organisms and identified shared properties not found in their relatives that lack this trait, that we can separate the species-specific features from the fundamental mechanisms subserving that trait, yielding meaningful biological insight into the core genomic features associated with this trait of interest. Fascinatingly, research in vocal learning, grounded by comparisons between humans and songbirds especially, has revealed that the principle of convergence applies on multiple levels. On a behavioral level, baby songbirds and humans both progress through a babbling phase with critical periods of neuroplasticity where learning must occur. But this vocal learning trait is also reflected in the neuroanatomy itself, with specialized forebrain circuitry and corticospinal connections associated with fine motor control that are found in vocal learning organisms, but not in their close relatives. Our past work has shown that even the genes expressed in these specialized brain areas themselves show convergent patterns of expression, with genes being differentially regulated in vocal learners in a way that they aren't in their vocal non-learning cousins. We're now seeking to extend these analyses to the epigenomic level of gene regulation, with the goal in mind of identifying the regulatory sequences that orchestrate gene expression where evolution is likely occurring. We opted to use ataxi, an exciting new technology that allows one to identify all open chromatin regions of the genome where transcription factor binding for gene regulation might be occurring. So in order to understand the evolution of vocal learning, we sought both to compare vocal learners like our friend Charles Darwin here with their non-learning vocal learning outgroups, non-human primates. But in order to harness that power of convergent evolution that we discussed earlier, we also sought to compare humans with vocal learning bats. So the rhesus macaque, as well as being a vocal non-learner and thus providing a baseline for human comparisons, also has brain regions associated with fine motor control related to hand movements. Whereas certain subregions of the hand motor cortex are associated with more crude grasping movements, other areas are associated with more fine motor dexterous movements, which in humans, of course, have been elaborated further to enable complex tool usage. By comparing these two complementary fine motor regions, we sought to identify what is common as well as what is unique to each. Furthermore, in bats, ample evidence is emerging that many species within this diverse order are also vocal learners, just like humans and songbirds. And in collaboration with Michael Yartsev, I sought to extend what we've learned from vocal learning birds to this promising new model for vocal learning in mammals. So in the macaque, I performed a tax-seq to epigenomically profile a suite of 14 tissues, including many brain regions related to vocal behavior and dexterity, including multiple subregions of the primary motor cortex. In the Egyptian fruit bat, likewise, I compared a tax-seq in a complementary set of 10 tissues, including regions that appear to be specialized in these animals for the production of learned vocalizations. These data are of high quality, as shown by these periodicity plots at left that reflect the regular spacing of nucleosomes and open chromatin regions. At right, comparing publicly available human data to the data that our lab has collected from bat, macaque, rat, and mouse, we can see that conserved open chromatin patterns emerge, with some differences that are worth exploring in individual species. To our great excitement, these data make biological sense as well. Comparison even with these highly specialized subregions of motor cortex, here between the hand control and the orofacial control region of macaque motor cortex, we see not just random sets of genes, but rather an enrichment of genes with known involvement in fine motor behavior. All told, this has resulted in a massive multi-species, multi-tissue epigenomic atlas that, as I will show you, has massive potential to expand our understanding of the biology of the evolution of the brain, especially the brain's capacity for fine motor behavior. And so what can we learn from these tissue-specific multi-species epigenomic data sets? First of all, that comparing these enhancer sequences across species allows us to identify both species and clade-specific evolutionary innovations. So by comparing epigenomic data across species, we've been able to identify lineage-specific as well as species-specific differences in regulatory evolutionary innovations. Whereas motor cortex regulatory activity is largely conserved among primates, we did find in humans that there are over 13,000 regulatory peaks that are active in the human motor cortex alone. Examining just one of these in more detail, we identified a previously unknown regulatory enhancer within the FOXP2 gene, a gene that has long been appreciated for its role in speech and vocal learning in humans as well as songbirds. We're currently expanding this cross-species work into the single domain as well, with comparable motor cortical data sets our lab has collected now being analyzed to connect those species-specific epigenomic differences to the specific cell types within the brain that they're active in. Furthermore, we can use these comparative epigenomic data sets to predict gene regulatory activity using machine learning. And a talented postdoc in our group, Irene Kaplow, has been developing convolutional neural networks in order to take all these primary data that I've generated and learn certain features of the sequences in order to predict the tissue-specific activity in other species where we haven't collected primary data. And this could be invaluable for the large number of extinct or endangered species that we're seeing every year. Furthermore, I'm also developing ways to functionally test and experimentally validate these sequences with massively parallel reporter assays, where essentially we can take these sequences that we've identified computationally and put them into a viral construct where then we can test the actual ability of these sequences themselves to drive expression as measured through RNA barcodes that we then sequence from any tissue we like. So in summary, this comparative neurogenomics approach reveals fundamental molecular and genomic mechanisms that underlie complex behavior. And these multi-species, multi-omics data sets provide a window into the evolution of brain circuits for fine motor behavior in mammals, as well as birds. And finally, that complementary computational and experimental approaches enable the development of testable models for connecting epigenotypes to behavioral phenotypes. And with that, I'd love to thank you for your attention, and I suppose I'll take any questions.
Hello, everyone. My name is Maribe Gamboa. I am working in Ehime University in Japan. And I'm going to talk to you about gene expression changes on multiple species associated with the latitudinal gradient. Gene expression analysis has been studied widely along the latitudinal gradient because latitude is associated with UV temperature and humidity variation. Previous studies have found population divergence, differential gene expression in multiple organs, and also parallel adaptation of latitudinal differential genes. And overall conclusions, genetic responses was associated with the latitudinal difference. However, multiple species studies have not been conducted, and this is one of my motivations. So I wanted to see the latitudinal influence, the gene expression difference of the species to observe a special selection. Therefore, I study transcriptional responses, gene expression diversity, and common molecular mechanism among the species. My target species is stonefly or Plecoptera, is aquatic insect. They live under the water, and they have been, they are food for fishes, and they are important because they're one of the aquatic insects most sensitive to environmental changes. They are worldwide, but they have high diversity at higher latitudes. In my previous study, I found that genome-wide analysis, they show a population divergence associated with certain environmental conditions. So to study the latitudinal gradient, I sampled natural population from the north to the south of Japan in 4 different geographical regions, and I selected 7 different species that they have different behavior and different habitat preference. So I extract the RNA, make RNA-seq libraries, and I conducted the bioinformatic analysis needed, and I obtained more than 3,000 genes and 101 species-specific genes. So now let's look about differential expression genes among within the species. So in this slide here, I show you a map of Japan and 4 different geographical regions that I sample in 4 different colors. And you can see the heat map of the differential expressed genes, and over the graphic, you can see the percentage of gene similarity. I obtained 637 differential expressed genes, and if we observe the 2 extremes, we can see that the higher latitudes, we found high number of species-specific genes, and the gene similarity was around 18%. And this gene similarity is increasing when we decrease the latitude. If we observe the expression diversity was highly associated with the latitude. So that could be one of the explanations. Again, if we observe the 2 extremes, I found that species-specific responses were at a higher latitude. However, and this was widely studied. However, I found lower expression diversity and high similarity among the species, and could be because environmental stress. And this one was observed before in fishes because of high temperature, and also in trees because of the drought. And to study the co-expression among the species, I analyzed co-expression. I did co-expression analysis, and I obtained 22 co-expression genes highly associated with the latitude. And these 22 genes clustering the species in 4 different groups, which is I expected. And the groups were associated with the 4 geographical regions. If we observe these 2 regions, in the differential expression patterns among the species were highly diverse, and could be the consequences of high species diversity or high protein diversity in these 2 regions. The heat map of these 22 genes is showing in the right side, and then you can see respiration metabolic genes. They show a high, were upregulated in the higher latitude, and this could be a consequence of latitude variation. The opposite pattern was in the developmental one, with hexamerine was downregulated at higher latitude. These community co-expression genes show a latitudinal clay that could be associated with the adaptation, and this was found in population studies in latitudinal valleys. So in conclusion, I found that the latitude is actually influenced in the gene expression pattern. So thanks to my findings, collaboration, and you for listening.
Hi, everyone. Thank you. Those were some great talks. I really appreciate it. We have a couple of questions. Hopefully we'll find a question for each one of you. Marco, you get a question from Oliver Ryder. Can the inference made from chromatin conformation be used to identify homologs across species in absence of karyotype data?
Thank you, Oliver. That's a really nice, interesting question. So when we start doing this project, we detected that each chromosome has a specific pattern. So of course, we wonder whether this is a characteristic of every species, and whether we can identify homologs with this kind of approach. And since through the EBP and all the genomic sequence projects, a lot of genomes that are going to be created, and the karyotype data is not going to be available for all the species through a standard cytogenetic meaning. Of course, a new method to identify karyotype will be important. So I guess that something is that we can do, and something that we are working on, is a pipeline to use eigenvector and chromatin pattern to identify homologs in the same of species of the same families, but also whether we can identify species and karyotype of different families will be something that we'll be answering like in the following period. So I think so. Thank you.
Great. Matthew, it seems regions of high divergence are rather localized in sympatry. Do you know anything about structural variants in these regions?
Yeah, thank you for a very interesting question. So that's obviously something that we've thought about. There are these long blocks in sympatry which seem to localize with centromeres and reminiscent of having sort of long fixed haplotypes. We've wondered whether those represent structural variants and searched for evidence of that, but we haven't found any. So we think that they're probably not structural variants and just the product of ongoing link selection.
Great. Peter Holland asks Shigehiro a question. Could you speculate on the possible functional consequence of the radically altered HOX-C cluster in sharks?
Well, it's quite mysterious. I mean, genome structurally speaking, I mean, the HOX-C genes, escaped from the constraint, but in terms of the temporal and then spatial regulation of the expression, they have maintained, typical HOX-like pattern. So in the end, maybe there's no huge impact in the expression, but the cluster structure underwent quite dynamic, escaped from the constraint. We are still working on this question. Thanks for asking.
Great. A question for Garima. Do you know which metabolites are produced by the gene cluster specific to the cold ecotype?
Thank you very much. That's a very nice question. Unfortunately, we do not know if there is a metabolite that is produced by the cold specific ecotype. We do have an estimate based on the similarity of the core PKS gene of this cluster. And it seems like it might be involved in the synthesis of either a pigment or a compound that is involved in UV or stress-related tolerance. But that's just an estimate based on the similarity of the core genes. And in lichen-forming fungi, in general, the compound and the clusters have not been connected. So, for instance, we know in Lazalia, in Umbilicaria postulata, in my system, we have 2 to 3 compounds, and we know that we have about 20 clusters, but we could not connect any compound to the genes. And also, we did not find any compound which was specific to the cold ecotype, so that we could then connect this to the cluster which is present only in the cold ecotype. So no, we don't know which compound might be synthesized by this cluster.
Great. For Maribeth, could the findings of your work be used to help diagnose environmental pressures?
Yeah, thank you for the question. Yeah, actually, it can help, but it can also give you some signs about local adaptation and also climatic change influence in the genetic and the gene expression divergence. Thank you for the question.
Right. And I actually have a question for Morgan. And you showed that you're going on with MPRA assays to look for the functionality of your variants that you've detected. Do you worry that looking at primate-specific variants in the mouse model could cause bias in the results?
That is a great question. And so sort of the assumption here is that the cis mechanisms, the promoters and enhancers, are likely where most of the changes are happening. Whereas in the transcription factors, the transacting elements, those are less likely to be changing. I mean, we saw this other beautiful talk showing, the strong conservation of Hox genes. But in general, we find that these transcription factors are so critical and, reused in so many different tissues that you're a lot less likely to alter those genes themselves. So it is an assumption that we believe that the sort of transcription factor milieu is largely conserved across species, but of course it's possible that there are some changes. Some transcription factors like zinc fingers do evolve a little faster than the Hox genes, for instance. And so, you really have to take it family by family when interpreting your findings. But yeah, I think for the most part, it's going to be the sequences themselves and comparing, different species variants in one species that's going to give us insights here.
Excellent. So I want to thank the speakers again for a really diverse and interesting session. And thank you very much. I wish I could give you an applause that you could hear. So
Thank you.
Thank you.